seminar. Today we're going to hear from Andrew Webb. Andrew is that rare beast, a biologist who uses technology to answer questions. And Andrew began his career, he did a PhD at Monash with a great deal of time up here at the University of Melbourne Microbiology. He was a C.J. Martin Fellow then at Imperial College and back at Bio 21. He joined WEHI and the Proteomics lab about 3 years ago. He has been, together with the team in the Proteomics lab, responsible for the rebuilding and rejuvenation of this lab. And I'm very much looking forward to hearing his talk today.
Thank you, and thank you, Liam, and thanks very much for the opportunity to share some of my work today. And as Liam mentioned, I've been working in the Proteomics lab here at WEHI for the last 3 years, and it's been quite eventful, and it's been a really good time. And today I'd really like just to present some examples of protein signaling mechanisms that I think will really illustrate just how proteomics can be useful in quite a range of different biological applications. However, I think for all its potential, mass spectrometry-based proteomics still remains a relatively underutilised tool, and that it hasn't really been incorporated enough into more biological-focused labs. So a few years ago, this really prompted this question, is mass spectrometry ready for the big time? And I think what they're really asking here is that given the advancements in the field, is it ready to become a more commonplace tool rather than just a specialty? So my answer to this is absolutely. It's an incredibly versatile tool that's particularly good at understanding protein functions and really, digging into disease mechanisms. So why isn't it being used more? In short, the instruments are quite expensive and really challenging to use. While the technology has improved enormously over the last few years, the usability of these instruments really hasn't. So unfortunately, most of the advanced proteomic technologies and capabilities have really remained inaccessible to a lot of the broader research community. And I think this is what our aim is really at the Proteomics Lab, is to really try and bridge this gap effectively and become embedded into research groups around WEHI to implement and standardise a number of more advanced proteomic research areas. So these areas now cover quite a diverse range of applications and include protein structure, dynamics, protein-protein interactions and their modifications. However, our core focus really revolves around protein quantitation, as the current instruments that we use can provide some pretty or very high quality quantitative data. So these areas can be further described by the types of projects that we run in each of these. And it's important to point out here that all these methods require individually tailored workflows and bioinformatic pipelines. But overall, they really reflect the ongoing work that's happening in the Proteomics Lab here at WEHI, and we've now worked with, I think, 21 labs over 8 different divisions over the last few years. So while it would be impossible to sort of cover everything today in this forum, I'm going to focus on 3 recent projects that I think clearly demonstrate what we can achieve with mass spec. So these will cover protein ubiquitination in the TNF signaling pathway. They will cover identification of a novel post-translational modification that prevents death receptor signaling during bacterial infection. And lastly, I'd just like to talk about a modified method for identifying interacting proteins that we've been developing over the last year or so, and I'll be using the SOX5 protein as the example here. So I'd also like to share with you today some of the very preliminary work that we're beginning on intact protein profiling, which I think is a direction that the field is moving towards in the near future. So whilst I'll be covering quite a bit of different biology today, I think the one central theme really tying this all together is the underlying development and application of the technology that really allows us to push our knowledge in these fields. But first, I'd just like to give a quick overview of proteomics, and in particular to demonstrate why I think proteomics is slowly becoming a much more important field. So I believe that phenotypes are described by their protein function. Whilst we all know that DNA contains all the instructions necessary for creating an organism, we're now beginning to understand the complexity as to why the genotype and phenotype don't always line up as closely as we used to think. And recent in-depth proteomics experiments have demonstrated there's only about 19,000 coding genes in humans. And while in individual cell types and cells within tissue, we only see about 13,000 proteins. So when we combine the discovery of ever more regulatory events between transcription and translation. And we combine this with a large number of reported, or identified or known post-translational modifications. The complexity inside cells becomes quite astronomical, and with some groups now estimating that there are 1 million different proteoforms. And by this I mean protein forms. So while we annotate genes and we have 13,000 annotated genes, we believe that when you include all their modifications, which potentially allow them to have different functions, even with cleavages as well, that we now have this sort of complexity, or we expect this sort of complexity with inside cells. And for quite a few diseases, this genetic characterisation alone can be very powerful. Genetic characterisation alone can be very powerful in identifying a phenotype. There appears to be ever more situations which are much more complex and indirect, and I think it's as a result of this. And this is where I believe the power of mass spectrometry really lies, in being able to sort of dig into and really characterise the mechanisms of different phenotypes. So historically, the most in-depth way of trying to understand disease phenotypes has been to use quantitative transcriptomics. And it's becoming quite apparent with the aid of high-throughput quantitative proteomics that transcripts aren't actually that good at predicting protein abundance. So this is a recent example from Matthias Selbach's group, where they measured absolute abundance and half-life of both mRNA and protein. And what they showed was that proteins are on the order of 5 times more stable and 2,800 times more abundant than their corresponding transcript. So while there's an apparent correlation with their copy number, it would appear that using transcript quantitation to infer and explain protein abundance is probably a poor substitute. So taken together, this huge complexity in the proteome that I was talking about in the last slide, and the lack of accurate correlation between mRNA and their protein, I think this really suggests that we're in need of some new tools and methods to really further understand the mechanisms underlying different phenotypes and different biological mechanisms. So in thinking about our ability to measure and quantitate changes in both transcripts and proteins, this has only really been facilitated by the advancements in the technology over the past decade or so. And while gathering global sequence information on DNA and quantitative information on transcripts it's becoming much more routine thanks to its next-gen sequencing, global protein analysis still poses a much tougher analytical challenge. So over the past decade, we've seen some pretty amazing advances in sequencing technology. And what you may not be as aware of is that we've been witness to some pretty immense improvements in mass spectrometry as well. And particularly with this is not just the instrumentations, but the sample preparation and the computational analysis that we do. So whilst sequencing and analysing a protein just about 15 years ago was quite a challenge, now we're at a point where in a single experiment we can identify and quantitate most of the proteome within a day. And it wasn't really that long ago, in the late 1980s, that John Fenn, he developed electrospray ionisation for peptides and proteins, which has really now become sort of the foundation of what we do in the lab here. Having said this, I believe that while this looks a bit exponential, that this really is just early days for proteomics, and I think there's a lot of development and advances yet ahead of us. And to take a guess, I think we'd probably be around this region. So before next-gen sequencing really take off, I think we're sort of just hitting the possibility of sort of next-gen proteomics. So it really is quite an exciting area for us in this area of research. So to analyse and interrogate different aspects of proteins, we first extract proteins, generally with detergents. We can enrich or separate populations at this stage, or we can just process the samples directly with a protease. And we typically use trypsin, as this provides some favourable properties on the peptides, allowing us to have a charge on the C-terminus, so it aids in sequencing. Digestion of the full proteomes, though, they generate an enormous amount of peptides. So we have on the order of hundreds of thousands of peptides that is not directly compatible with mass spectrometry. So therefore, the first step in our generalised workflow is to reduce this sample complexity by either pre-fractionating with a variety of different methods, or enriching for populations that we really want to look at. So whether we want to look at phosphorylation from titanium dioxide enrichment, or using antibodies for a host of other different types of modifications. So these processed samples are then run on an LCMS, and their Tandem spectra is acquired. And by Tandem spectra, I mean that the peptide ion is measured, it is then isolated in the instrument and applied energy to or fragmented so that we can derive some sequence information. So this information is then processed to yield identifications and quantitative information. And the types of analysis that we do really just dictate on what the experiment was that we were designing in the first place. And obviously you can see from here there's an enormous variety of ways that we can feed these instruments. And the final point that I just wanted to make on this slide is that the information that we get is highly quantitative. So with our newer instruments, we have on the order of 5 orders of magnitude. And we know that within cells, proteins have a dynamic range of around or a bit over 6 orders, possibly up to 7 orders of dynamic range. So this brings me to the first of the 3 projects that I'd like to share with you today. And this is a project that I worked on just prior to joining WEHI and involves characterising ubiquitination events following TNF stimulation. This work was published in Nature, and I think it's a great example of how we can use mass spectrometry to really delineate and interrogate ubiquitin signalling systems and ubiquitin signalling within signalling cascades. But just before I move on and talk about that, I'd just like to introduce you to ubiquitination, as I'm about to show you, is crucial for maintaining appropriate signalling in the TNF receptor signalling pathway. So ubiquitin is an 8.5 kilodalton protein that's one of the most conserved proteins known. During ubiquitination, ubiquitin is covalently linked to the lysine of a substrate via an E2 and E3 ligase complex. And ubiquitin chains form when subsequent ubiquitin molecules are added, and these can be extended to get quite long chains of ubiquitin. And as I'm sure quite a few are already aware, there's sort of 2 main types that are known or accepted. That we have these K63 link ubiquitin linkages that are involved in molecular assemblies as scaffolds and that allow recruitment of other proteins that can then potentiate a signal. And also K48 linked chains that target proteins for degradation via the proteasome. However, ubiquitin has 6 additional linkages that can form, but relatively little is known in comparison about these unconventional chain linkages. And we've also recently observed, this is some work from Jared Sandow, where we recently observed all these linkages. You can't see the K29 because the instrument wasn't set up correctly at the time, so we missed out because it's quite a small peptide. But we can see all the others, demonstrating that I think that these linkages are likely to be physiologically relevant. And for 5 of these linkages, the structures have actually been solved. And I think these clearly demonstrate that these ubiquitin, or so far, that these ubiquitin linkages fall into 2 distinct categories. We have this more closed, compact conformation, albeit with some small structural differences, that we know that K11 and K48 signal for degradation, while we know that these K63 linkages are in a much more extended conformation. And these include the new MET1, or these are the linear ubiquitin peptides where you get this end-to-end conjugation. So the N terminus of ubiquitin fused to the C terminus of the next molecule. And we know that these molecules, being in a much more open extended conformation, that they're much more accessible to the different ubiquitin binding domains that we know exist. So as I alluded to earlier, ubiquitination is crucial for TNF signalling, and it's already known that to involve more than one of these chain types. So as you're well aware, the role of TNF the role of TNF in inflammatory disease is really well established, and that inhibitors of TNF are very good at treating diseases such as psoriasis and rheumatoid arthritis and Crohn's disease. And that the biological drugs targeting these now currently generate some $30 billion a year, $30 billion a year in revenue. So nevertheless, even with more than 30 years of study, as I'm about to show you, I think our understanding of this signalling pathway is certainly not complete. So this work began when I was working at a postdoc in Imperial College in London, where I was working alongside Henning Walzak, who was collaborating with John Silk, who was out at La Trobe at the time. And it was in Henning's lab that Tobias Haas, a postdoc there at Imperial, asked the question of the TNF signalling complex, is this it? And set about seeing if there was any unknown components. So to do this, he generated a tandem affinity tagged version of the TNF molecule and used it to pull down the entire signaling complex. So by mass spectrometry, we were able to identify all the known components of the TNF signaling receptor signaling complex, and we're able to identify 3 new ones. So Hoyp and Hoyle, these were first identified and published while I was in the lab, but I became involved in characterising SHARPIN's role in this complex. So we now know that these 3 new molecules, or 3 new proteins in the receptor signaling complex, form a trimeric E3 ligase complex known as the linear ubiquitin assembly complex, or LUBAC. And we know, so obviously, that these mediate these linear ubiquitin linkages. So work in Henning's lab had shown that this complex is recruited to the TNF receptor signaling complex in a TRAD, TRAF2 and CI dependent manner. And it was required for downstream NF-kappa B and junk activation, and also its presence inhibits cell death induced by TNF. And as good luck would have it, not for the mouse, but there was a mutant mouse strain that presented with a severe chronic proliferative dermatitis, which was effectively a SHARPIN knockout. It had a spontaneous base pair deletion. And the association of the skin type in these mice with TNF signaling was further supported by crossing these onto TNF knockouts, so that one loss of an allele was enough to restore the skin phenotype in these mice. So we now knew that this phenotype was really dependent on TNF signaling and potentially through some linear ubiquitination events. So we now know, of course, from the really nice work from James, Joanne, John, and Motti, who presented this work at a Wednesday seminar recently, that the skin phenotype that we were observing was likely a result of TNF activating the regulated necrosis pathway. And they really demonstrated really nicely in this work that it was actually being regulated through RIP1, which now appears to be this central gatekeeper or regulatory checkpoint for cell fate after TNF stimulation. But this still leaves us with a few questions as to what is LUBAC's role in this complex. What we're really asking here is, I guess, what are its substrates and what effect is it having? And as we know that RIP1 is a central gatekeeper to TNF signaling, is RIP potentially more involved in this, given that we know that it's extensively ubiquitinated already? So to look at this more specifically, we needed a way of being able to detect linear ubiquitin chains in the pathway. And while there are antibodies to do this, they generally aren't very good at discriminating between all the different possible linkage types. So how do we detect all 8 linkages of ubiquitin with mass spectrometry? Well, a nice quirk of nature, the arginine at the third last position here in ubiquitin provides a nice triptych cleavage site. So when the ubiquitin molecule is conjugated to its substrate via its C-terminus, or we have a K63 linked ubiquitin chain or a K48 linked chain, when we digest these with trypsin, they generate peptides that represent all the known linkages that can exist between ubiquitin molecules. So we can then tell the mass spectrometer to target these and also quantitate them. So in this instance, I used a targeted mass spec approach. I used a triple quad instrument. It was actually a Q-trap instrument, where we actually we measure the area under the curve of the fragment ions. And while this normally gives us specificity in really complex samples, the reason I used it in this instance was because it was by far the most sensitive instrument we had at the time. Our newer instruments are more sensitive to this again now. So now that we had a way to target these linkage-specific peptides, in addition to the peptides from the rest of the complex, so not just the ubiquitin ones, but also what else is there, we needed a strategy to really interrogate these different protein forms. Because if we were to digest all these together, I mean, yes, we could identify the linkages, but we had no way of linking which linear chain or which chain was coming from which protein. So to do this, I combined that TAP-TNF approach that I mentioned previously, and then we started running some 2D gels to really separate proteins out into their individual protein forms. Just to give you the idea of a scale of this sort of experiment, we were using, in each pull down, we were using about 10 to the 10 cells, and we're only generating sort of a few micrograms of protein. And we were having to use a dye, a fluorescent dye, to be able to detect these proteins. And I was typhoon imaging and using a robot to pick these spots. So I think I ended up running about 10 gels from about 10 preps for 10 to the 10 cells. I was cutting about 100 to 200 spots out of these gels each time, manually digesting them and then running them on about a 30 or 40 minute gradient. So this is really not a high throughput approach. So in the end, I was able to generate some really clean hits in both RIP and NEMO as substrates of the SHARPIN containing LUBAC complex, detecting linear ubiquitin peptides in both of these. You can see these little peaks here represent detection. And additionally, within the RIP spot, I was able to see within a single form of RIP spot, so by a single spot, so there's a single proteoform of RIP, I was detecting not just linear, but also K63, K48, and K11. So the molecular weight of RIP here is up at around 250 kilodalton, which would mean that there's around 20 to 25 ubiquitin molecules attached to RIP. And NEMO, in this case, was running at a molecular weight consistent with 2 ubiquitin molecules being attached. So 2 ubiquitin molecules with a single linear chain between them, or linkage between them. So then we took this another step further and looking at overexpressed MIC-tagged RIP, in which we could better enrich for the ubiquitinated RIP and interrogate it more closely. And in this case, we compared reconstituting with wild-type HOIP and also a mutant form of HOIP that can't mediate these linear chains. And by cutting the gel bands up the molecular weight region, we could infer the linkage usage as these chains were extending. So these bar charts represent the intensities of the linkages that I was detecting as we're moving up the gel. And so what you can clearly see from this is that it's getting very heterogeneous very quickly. And when we have a mutant HOIP that can't mediate these linear ubiquitins, the linkage linear chains, it appears that we almost have a corresponding replacement for linear with K48 linkages, which we don't know are biologically relevant or not yet. What we can also tell is that it appears that these chains are beginning with a K63 linkage before they become much more heterogeneous. And it becomes impossible to resolve any structural information out of this, but given that we only detected a very few individual spots from a 2D gel at that really high molecular weight region, really suggests to me that we're actually probably looking at quite a highly ordered structure. It's actually interesting to speculate about the possibility of such a structure. So given that we had, on average, about 20 ubiquitin molecules, and that we were detecting 4 possible linkages, the maths are quite simple, that we had this structure contains over 1 trillion different possible combinations. This is from a single PTM mediating an extraordinary amount of complexity. So I think given the level of the complexity that could arise, that I think this process has to be exquisitely regulated. So just to summarise this part, the first part is we've shown that the LUBAC complex is recruited to the TNF receptor signaling complex and that it linear ubiquitinates both NEMO and more extensively RIP. So we also now know that NEMO, it has a ubiquitin binding domain that actually has a 100-fold higher affinity for linear ubiquitin chains than it does K63. So we think that this most likely amplifies its recruitment and really drives for cell signaling and cell survival through the canonical NF-kappa pathway by providing a scaffold here for it to really bind to and amplify. However, when we lose SHARPIN and we get a corresponding loss of linear ubiquitin chains, this really results in RIP1 activating RIPK3 and MLKL to generate regulated necrosis or necroptosis. So there's still a question that remains for me with RIP1 is how does it actually decide which cell fate to choose upon TNF stimulation in a physiological setting? So whilst we know from John and Modi's recent work and some of the other recent work from GSK and Genentech, where they know that the RIP1's kinase activity looks like it's important in this process, I think it's possible that upstream of that these ubiquitination events are possibly controlling this. And given that the antibodies that we have to these chains aren't very good, I think mass spectrometry is probably the only way that we can really address this accurately. So this just brings me to the second part of the talk that the second project that I'd like to talk to you about today. And this project isn't too big a jump away from TNF signaling, but involves determining the mechanism for blocking cell death by an effector protein from E. coli. So in this project, we identified a new type of post-translational modification that was specifically blocking cell death receptor signaling, and we published this in Nature late last year. So this project involved a larger group from WEHI, but it was predominantly led by Liz Hartland over at the Peter Doherty Institute and really driven by Jacqueline Pearson, who was a PhD student, a very talented PhD student at the time, has now moved on to a postdoc in Liz's lab. And given that Liz presented this work recently very elegantly at a Wednesday seminar, I'd just like to give you a brief overview as I'd like to talk a little bit more about the strategy for identifying this modification as this sort of ties into our push in the lab of looking at the proteoforms of proteins, starting to look at things intact rather than trying to break them up into more manageable bits with trypsin. So briefly, enteropathogenic E. coli, they utilise a type 3 secretion system to manipulate their host environment and promote adherence as well as interference with the antimicrobial host response. And they do this by injecting from 50 up to 100 effector proteins, depending on the strain of the bug. And in a yeast-2 hybrid experiment, Jacqueline was able to identify that one of these proteins, one of these effector proteins from E. coli called NLEB, bound to Fad, and actually the death domain of Fad. And when she ectopically expressed NLEB, or it was injected by the bacteria, she'd shown that this could block formation of the TNF-induced canonical death-inducing signaling complex, or DISC, and also prevent the FAS induction of this complex as well. And this really, the subsequent proteolytic activation of caspase 8 was also blocked in this case. And it was also shown prior to this work that NLEB from C. rodentium had also been previously, or been observed to be an O-linked, or observed to have O-linked glycosyltransferase activity. So we had quite a head start in trying to identify sort of what type of modification NLEB was really performing on the death domain of Fad. So to get an idea of the full extent of the modification, we thought it would be important to combine two approaches. The first being a top-down approach, as it's only by looking at the intact protein as a whole that we can get an idea of its exact mass and of the protein, as well as any modifications that are present. And in this case, we have some advantages in that we can see if there's any degradation products or any sequence variants. We pick that up because we're measuring the whole intact mass of the protein on the instrument. And when combined with a more traditional bottom-up approach, where we digest things with typically trypsin, we can really hone in on and try and detect the site of the modification. So while using this approach alone, which is what we do for 95% of the time in the lab at the moment, we do identify a good portion of the protein, but there is an opportunity, there's a chance that we may miss all of everything that's going on here. So in essence, this really became a top-down informed bottom-up analysis. So when NLEB was reacted with Fad and a range of different sugars, we detected a complete conversion to a heavier mass. So what you're looking at now, this is the raw result of the intact protein flying on the mass spec. When peptides ionize, they generally come out in one or two different charge states because we're measuring mass overcharge. With proteins, when they ionize, they typically ionize in multiple charge states, so we detect them multiple times on the instrument. And it actually becomes quite nice because our mass accuracy improves because we can use all these peaks together. So the mass difference, so here I've got Fad in black, and then the modified, I've already got it listed here. I didn't realise I had it there. The mass shift between these two equaled 203 Daltons, and this corresponded to a single addition of a glucanac molecule. So this clearly demonstrated to us that we were just chasing a single modification on the death site, the death domain of Fad. So by doing an in-gel digest, we're able to pinpoint this site of the modification. And what really shocked us when we did this was that glucanac turned out to be N-linked, which wouldn't be such a shock, but it was on an arginine residue, as opposed to an O-linked, which would have potentially been on this serine residue. So there's the added benefit in this case of being N-linked in that they're much more stable on the instrument, and when we break this apart and we fragment it, we actually were able to retain this side chain modification in some of the fragment ions. So we had a nice way of sort of unambiguously characterising where this modification was. So we were the first people to identify this modification, which is, I think, what really led to it being accepted in Nature in the end. And so the next project that I wanted to tell you about takes us away from death receptor signalling and involves a technique that makes the most of the quantitative ability of mass spectrometry. So this is a modified approach that I've been developing over the past 12 months or so with the aim of more clearly identifying protein-protein interactions. So this is generally something that it's quite hit and miss, and it's very difficult to get this working properly. And there's a few reasons to that. And so in order to enrich proteins, we generally use a solid support in conjunction with either an affinity reagent or antibodies to enrich our target proteins of interest. And there's two main factors that really determine how well these pull downs work. And the first being time, in which we lose a lot of our lower specific interacting proteins. And the second is the level of contamination that we see. So whether we're digging deep enough and being able to filter these out from all the noise. And we can clearly see a good example of this, and this is from the TNF, the Ubiquitin story that I was telling you about, where we typically have about 90 to 95% of the protein that's here, and from a single step, this is a flag IP. This is 95% of this is non-specific proteins. And in this case, we really had to resort to the Tandem step to do the Ubiquitin work, where we had a sequential FC affinity step. But having this step really blows out our time, so we get killed on identifying sort of the lower abundant, lower affinity proteins here. And what's also quite apparent from this work was that Western blots can be really misleading when you're doing pull-down experiments, and this is something that I get hassled by quite routinely. The Western's working, but the pull-down's not. So a recent example of this technique that we're developing comes from some work that's generated by Edmund Linessy, who is a PhD student that I'm co-supervising with Sandra Nicholson and Nick Nicolai. And a part of his project is directed at identifying a physiological role for the SOX5 protein. As I'm sure you're all quite familiar with the SOX family of proteins, I'll keep this introduction brief, but SOX 1, 2 and 3, they've been extensively characterised and have quite an overt phenotype. SOX5, however, there's very little known about its physiological role. So SOX5 contains an SH2 and a SOX box binding domain, a SOX box domain, that are characteristic of the SOX family of proteins. However, it has a much longer, or a considerably longer N-terminal domain that's known to be relatively unstructured. So one of the strategies that Ed is undertaking to really try and gain insights into the biology of SOX5 is to look for interacting partners. And in this instance, he's flag-tagged SOX5 and he's overexpressing it in 293 T cells. So given the homology of the SOX box to the other SOX proteins, we'd probably or likely expect the same sort of E3 ligase complexes that we know interact with the other SOX box of other SOX proteins. However, we're obviously much more interested in trying to identify what its substrates are. So using what would be considered a standard quantitative method to try and identify interacting proteins, where we plot, this is generally the summed peptide intensity of protein, or summed peptide intensities for each protein. We plot out the SOX5 IP, and on this axis we plot out a control IP, and we usually use this control IP to filter things out, to provide us with sort of specificity within the pull-down. So this is what the SOX5 pull-down looks like. However, we're left with around 380 proteins in this case that appear to be specific, and this is clearly a much larger number than we'd expect, and there's quite a few proteins in here that don't make much biological sense at all. So the challenge then really becomes to find out the true from false positives within this list. So when I was thinking about this and what we're really trying to achieve in these experiments, it occurred to me that we're really only going to be able to enrich for proteins out of the complex cell lysate. And there's always going to be contaminating proteins no matter how much we can try and clean this up or make this more efficient. And then a while ago, about a year ago, it clicked that what if the underlying contamination is mostly a function of its protein abundance in the lysate? So now what we've started doing routinely across all our pull-down experiments in the lab is to run the underlying lysate that we're pulling things out of. So this now provides us with a third axis that we can filter our data on. And even if our proteins are relatively high abundance, this is in the SOX5 IP, this is the lysate coming out on this axis and the vector IP. Even if things are relatively highly abundant, we can still characterise them as being likely to be specific. So typically, previously we would have probably omitted these and just assumed because they were high abundance, they're just there because they're non-specific and we just can't get rid of them enough. So now we really have a way of creating a much smaller, more manageable list. So we've now got 89 proteins or potentially interacting partners. And there's quite a few of them that now look really interesting. I'm not going to go into the biology of what's in this list. This is something that Ed's going to be pursuing, and he'll most likely be presenting next year at his final PhD seminar. What I did want to point out was, though, so this is just using a simple EMPAI calculation. So this is just the number of observed tryptic peptides over the number of observable tryptic peptides. And this just enables us to give us a sort of a ranking of protein abundance or relative protein abundance within this sample. And what you can clearly see here is we have SOX5 up here, we have this RNFF7, this is ring box 2, this is the uniprot gene annotation. TSEB1 and TSEB2 and colon 5. These TSEBs are actually the long and B and C. So we've basically recapitulated this whole complex in this IP. So I think this to me suggests that there's the potential hits in this list. While it's in an overexpressed 293 system, I think now Ed has really got some nice targets to try and chase. So while this is also working quite well, he's also got some really nice reagents to go forward with. So he's got some point mutations and truncations of this N-terminus domain. So using this technique, we can now really go in and seeing if this N-terminus domain is really providing any specificity for substrates that SOX5 might be acting on. So this brings me to the last part of my talk today, in which I'd just like to share with you some of the more exciting developments in our lab and in the field. And while I've presented some of the, the sort of the more peptide-based strategies that we've been using, it's important to bear in mind here that when we're quantifying proteins, that the identification that we make is generally an amalgamation of all the different protein forms that are there. And we tend to lose a lot of the contextual information and the finer details about the modification state and the various forms of that protein that might exist. So a way to address that is by omitting trypsin altogether and really just looking at the proteins themselves on the mass spec. And recently, this has been quite exciting for us in the lab, is that there's some more well-established mass spec groups, so from Joshua Kuhn and from Neil Kelleher's groups, that have reported some really nice strategies for quantitating complex mixtures of intact proteins. So Joshua's group is using a Silac-like approach. It's not quite Silac, but it uses a neutron-encoded mass and requires a very high-resolution instrument. And Neil Kelleher is pursuing the more traditional label-free approach. And this has clearly a huge benefit when we're measuring biological systems, in that it keeps all the contextual information of the proteins intact. So that, as I said, any modifications or any cleavage products, these can be observed and quantitated. So this is really not without its challenges, though, as proteins don't tend to behave as well as peptides. They're harder to separate, harder to get in the instrument. And it's really only been possible to start thinking about this in the last couple of years as we've had the instruments that have really developed and allowed us to do this with their sensitivity and their speed and their mass accuracy. So while these two groups have really been trying to identify everything they can in a complex mixture, we're starting to develop a slightly different approach here at WEHI. So we're planning on developing a combination of strategies around shotgun intact protein profiling. So we're certainly not going to be at the case where we can identify everything in a cell, but I think by looking at smaller populations, whether they're based on modifications or you're looking at protein-protein interactions, or maybe you want to look at a whole organelle or something like that, we'll be able to analyse this data and sorry, I'll go to the next slide. I just lost myself. So these will be run on an intact mode on the instrument. So we won't be trying to identify anything at this point, but we'll just be using the speed and the sensitivity of the instrument to actually profile everything that's coming off in the protein form. And it's in the analysis step that we'll go back and actually look and identify all the things that are changing between sample sets. Once we've identified the protein forms that are changing, we're going to use this as a target list to re-interrogate the sample. So we can re-inject the sample and then go in and analyse and look for the things that are really changing. And this has a bit of an added benefit as opposed to the previous group's approaches, in that we'll be able to spend a lot more time on these and really interrogate them fully. So we should be able to characterise these to quite a good extent. And I think the major benefit for me will come in looking at the thing that I've been becoming more interested in is this idea of PTM crosstalk, that we can identify multiple post-translational modifications on single proteins and how they potentially talk to each other within signalling pathways. So this is our first attempt on our brand new high-resolution Bruker-TOF. We've just had them commissioned. We've just been trained on them. It looks eerily like a 2D gel, but it's not until you look at these in 3D that you can clearly see the benefit of this type of approach in being able to individually measure these intact protein forms. So these, as I told you before about the proteins, they fly in different charge states. This is exactly what we're seeing here. So you're seeing multiple different forms. These are from 30 kilodalton proteins, we're detecting up to 60 and 70, 80 kilodalton proteins. This sample was actually just a test sample, but it was a depleted plasma serum. And we're actually quite surprised to see all these sharp spikes here. And these are a really complex mixture of high abundant but low molecular weight proteins that we typically, when we look at these samples, and we've looked at this sample in particular previously, we identify all these proteins and we get lots of peptide hits, but we miss all this because the way that we process the samples, we generally lose a lot of these small molecular weight proteins. So we think this is actually going to be a really nice way of looking at plasma complexity as well going forward. So within this sample, we detected around 200 different protein forms. And we're beginning to work with Brooker more closely to develop this technique. And they've recently informed us that on the same instrument that we have, that they're detecting around 1,000 different protein forms within a sort of a short 15-minute gradient. So I think on this timescale and this throughput, it really makes it feasible to start applying these techniques to some really interesting biological problems. So I'm also, and I just mentioned the Brooker. So I guess what I've presented today is a small snapshot of what we've really got going on in the lab. And there's plenty of other great projects that I'm particularly excited about. There's the surface profiling. I mean, these are projects that I would have liked to have talked about as well today. I think, hopefully these will come up for their own seminar with the researchers that are actually driving them. So I promised Jared that I'd plug the cell surface profiling. So Jared's been able to work up this strategy for looking at the surface proteins of cells to a point where he's now identifying between sort of 500 and 800 proteins quantitatively. And I think this sort of technique would be particularly useful for people looking to profile cell-sorted populations or cell populations where they're mediating an effect where they would expect a different protein changes on the surface of the cell. Then we're also pretty excited about some of the SILAC target ID work that we've been doing. So in particular with stuff we're doing with Pat Sharp and the chemical biology and the cell signaling division. And I do really believe that this is the early days for proteomics and that we have a good amount of development to go. And I hope that what you can take away from this is that proteomics is quite really a versatile tool that can really solve quite a range of different and intriguing biological questions. So we thrive on collaboration, so if you feel like you have stuff that you can work with us on, we'd be more than happy to have a chat with you. So this just leads me to my acknowledgements. So firstly, I'd just like to thank Giuseppe. He generated the data for the gluconac modification and that intact profiling you see. And he's really been instrumental in helping build the lab into what we can do and what we can achieve today. I'd also like to thank the rest of the Proteomics lab for their support and by making the Proteomics lab a pretty great place to work. I'd also like to thank the rest of the systems biology and personalised medicine division for their support, sort of bioinformatically and computationally. And I believe I've mentioned most of the people that did a lot of the work during of the projects that I've talked about, and also to the guys at Brooker. They've been incredibly helpful with sort of helping develop methods on the instrument and really helping us to push the boundaries of what we can do here. And lastly, I mean, while I could only present a snippet of what we've been working on, I think there's a lot of great work that's now coming out of our collaborations. And without you guys, we don't exist. So thank you for your support. And also thank you for your patience sometimes as well. These instruments can be temperamental, and until we had these new instruments arriving, we really only had sort of one high-end instrument, facilitating all this work that's going on. So I thank you for your patience, and thank you for listening.
Cells have a dynamic range of 1 to 10 to the 7.
Up to 10 to 7.
And the highest machine has a between 4 and less.
Yes.
At the moment. So presumably you are detecting the lower abundant proteins less likely to.
Absolutely. Yeah. We generally.
So do you have an example of something that you might know that is low abundant but you can detect it and something else that you know you can't detect?
Yeah, no, we definitely have lists of proteins that we know we're detecting. I mean, for those sorts of experiments where we're trying to really dig deep and quantify things that are very low abundant and have a very low copy number, we tend to pre-fractionate them quite heavily. So while we have, on the instrument we have maybe 1 to 2 times 10 to the 5 dynamic range, we can surpass that by loading multiple injections on the instrument. So we could run 96 fractions, it just blows out the mass spec time essentially. But I don't think doing global protein quant is not, we're not quite there yet. It's not something that I'd be recommending to do routinely. I think mass spec, the real power of it is interrogating smaller populations of things. So if you're wanting to look at the phosphoproteomics or you're wanting to look at protein-protein reactions, I think that's where we're hitting projects out of the park, and it's going really well.
comment on the sort of computational tools that are starting to emerge when we're getting these more and more sensitive instruments, because what we've found anyway is that they just lead to larger and larger sets of proteins, but that the computational tools that are available aren't keeping up with able to deal with these big sets and make sense of what's really going on. So is there any movement in that field to try and keep up with that sensitivity? And
Yeah, absolutely. It's a big issue. I mean, it's something that we're constantly fighting. I mean, there is a lot of development going on, but I mean, within the proteomics community, there's the proteomics community, and then there's really the bioinformatics community, and they are meeting and they're talking more. But there's an enormous amount of development to go yet, and particularly with that sort of stuff. I mean, doing sort of network analysis, and now that we're having these really huge complex data sets quantitatively, I mean, actually trying to tease out. I mean, I think there's enormous possibilities for. It'd be lovely. It'd be great to work with people that really were, fantastic and doing great stuff in network biology.
I think one of your earlier slides, the numbers suggested that you believe that there are 100 different molecular forms on average for every protein. Seems like a very large number.
It does.
What's the breakdown? How much of that is sort of...
Oh, it's a good question. I mean, these are just early estimates, and they're surely people, calculating these on how many within a cell, how many phosphorylation sites are we seeing, how many ubiquitination sites are we seeing, how many oxidized methionines. There's all these modifications. So as I said, there's 107 different known post-translational modifications that are happening in mammalian cells. And it really is just an estimate. It's a big number, and it's much bigger than we used to think.
Andrew, just looking at the SOX5 example that you showed. So you pointed out the proteins that were uniquely found in the SOX5 IP. How do you explain the proteins along the bottom axis that are uniquely identified in the control? Which, if I read it right, would have been a flag IP of
Yes.
cells that were transfected with vector only?
Yes, exactly.
I mean,
So these guys down here, you mean?
yeah.
Yeah, these are vector-specific effects, I think. These are the proteins that maybe there's a surface on the bead, on the M2 bead. They might be binding, I think TRIM21 has an antibody binding domain, or it binds, maybe it doesn't have a domain, but it binds antibodies with reasonable affinity. So I think the things that are more resin-specific that are coming down.
And wouldn't you expect to see those up here as well?
Yeah, yes,
The
definitely.
fact that they're unique in the control is what I don't understand.
This is done in triplicate, and with the instruments themselves, the way that they sample proteins can also be quite stochastic in a way. So that when they get to lower and lower abundant, we don't tend to ID the same things over and over. So it could just be that. You're right.
Andrew, of course, phosphorylation is the most common modification. Is it now very routine to be able to follow all the phosphorylation sites?
Yes, there are some labs that are doing it incredibly well, but it's quite a difficult process, and it's something that we've been working on. We're getting very close. But yeah, I mean, you generally, I think it's about 0.5% of a proteome that is phosphorylated. So when we digest, 99.5% of things that are there aren't phosphorylated. And we do have some really nice enrichment strategies that we can use to pull these out. So we use titanium hydroxide. And people are routinely reporting sort of, between 15 to 20,000 phosphorylation sites quantitatively in an experiment these days. So we aim to be there, and hopefully we'll be there soon. I've got a few projects, and Maria Tanza, we're sort of trying to develop our own in-house sort of strategies for doing it. So.
I might have misunderstood you when you were talking about whole protein analysis in that beautiful two-dimensional, three-dimensional thing you showed us. Is that telling you that at some stage we can bypass tryptic digestion of proteins and still understand their properties by using mass spec?
I think so, yeah. That's certainly what we're wanting to head towards. I think we're probably not going to be, with the current batch of instruments, we're not going to be quite there, but I think we're certainly starting to make inroads and sort of plan the development process. So by the time the instruments, these instruments, every year they come out with a newer, faster, more sensitive instrument. So you really got to stay on the forefront and keep developing, and with almost the expectation that the instruments are going to be a lot better in 3 or 4 years' time. But absolutely, I think it's going to be the case where doing things like protein-protein pull-downs, where we can analyse the intact proteins and look at what their modification. I mean, I'd love to do the TNF signaling pathway and look at things intact. I mean, obviously teasing out the linkage chains would be difficult, but looking at all those phosphorylation events and being able to do that temporally as well, I think it's going to be key to sort of understanding how a lot of these signalling systems actually, the mechanisms of these things actually work.
Hi Andrew, I'm Maggie, by the way.
Hi, Maggie.
So can you collaborate? I'm really intrigued with this concept of across models. Okay, so when we do genomics and transcriptomics, we work from a cell model into an animal model into humans. And so how much is being done across models to sort of look at proteins and how reproducible, I guess, it is across different models?
Yeah, I mean, a lot of the systems we use are cell-based. They're all in vitro cultures that we're doing these things on, and I don't know, it's a hard question to ask, a hard question to answer, but I think given that the level of technology of these instruments, we can start thinking about doing things in vivo. We don't need as much sample, we can get enough coverage with a small amount of sample.
I guess the reason I ask is if you have a really good mouse model for a neurodegenerative disease and you want to identify a biomarker, is it worth working in the mouse model, or is it worth going to CSF from patients, which is a bigger ask?
I don't know. I'm happy to go the in vivo model. I mean, a lot of what we do, because everything that we do is typically developmental, we always sort of sit down and we'll discuss projects, and then we'll say, okay, let's try this and try this and try this. And it's generally just a matter of sort of an iterative process of what's going to work best. So, sorry I don't have a better answer.
Okay, well, if there are no further questions, join me in thanking you.